Hi everyone, my name is Dr. Yusof from Leeds, United Kingdom. I'm reporting for Room Now for this year's EULAR 2021 Congress. I'm delighted to be joined by Dr. Panagiotis Garantziotis, who earlier presented his work as an oral presentation. The abstract number is OP0019. Hello, Dr. Panagiotis.
Hello. Thank you for your invitation.
You're welcome. Would you like to tell me a little bit of the background of the study and the objectives?
Yeah. As you know, I'm a physician. Also, I'm a physician with strong research-oriented educational background. I'm currently I'm receiving my internal medicine training at Hanover Medical School. Since I've completed my master thesis at the lab of Professor Dimitrios Boubas, I realized that I was truly interested in a career incorporating substantial research. That being the case, I made the decision pursuing a research-intensive career in of the physician scientist. Previously, I was being hosted as a EULAR fellow by Charité Rheumatology Clinic in Berlin, where I gained a strong, solid cross-disciplinary training in the field of translational medicine and more specific in the field of functional genomics. Currently, as you may have already noticed, I consider bioinformatic analysis of large-scale data quite challenging. Currently, we are working with my colleagues in the field in developing computational pipelines that may probably facilitate a more thorough stratification of patients with autoimmune diseases. I hope that our research may facilitate, may provide a solid basis for that may in turn assist personalized therapeutic decisions.
Yeah, very good. So thank you so much. Yeah, we definitely need a lot of bioinformatician and also machine learning. So regarding to your current abstract, would you like to describe what were the objective of the study?
You know, yeah. Also, the major goal of our study, as we said, it was to construct a computational pipeline, a translational platform that may allow a more thorough and more specific molecular stratification of patients with lupus, independently of their clinical annotation. So initially, to achieve that, we applied a co-expression analysis in whole blood transcriptomic data of patients of our publicly available data of patients with lupus. We recruited 120 patients with lupus. Taking a step forward, we used publicly available drug signatures of compounds that are either currently used in clinical practice or have failed in actually clinical trials. And we tried to identify compounds that may efficiently counteract, reverse the transcriptional signatures of patients with lupus. So initially, we used these molecular specific signatures of patients with lupus, and taking a step forward, we use this publicly available drug transcriptional data from drug databases in order to identify, in order to propose personalized therapeutic solutions. Taking a step forward, we tried to not only to identify compounds that are currently used or have failed in actually clinical trials and are and fit to specific molecular endotypes, but we also tried to identify novel compounds that FDA-approved compounds that may play a role in the treatment of patients with lupus. As you may, our study, of course, has certain limitations, but we are working with my colleagues in order to optimize our.
Okay, fantastic. So you're trying to define the molecular phenotypes and also the response to therapy. So just in a few words, can you please summarize the key findings of your study?
So as we identified five molecular endotypes of patients with lupus, and we tried in our study, we achieved to recapitulate all the pathophysiologic processes that are currently that have been discovered, have been reported in the literature. So initially we identified molecular endotypes of patients where neutrophilic deregulations plays a central role. We have identified molecular endotypes that are associated with type 1 and B cell responses. Additionally, another molecular endotypes mainly was associated with processes that were implicated in mRNA splicing and mitochondrial function, and we used and taking and in next step we found that bortezomib, a proteasome inhibitor which has been tested in clinical trials of patients with lupus, might be a promising therapeutic option for patients of group 4 of the neutrophilic class of patients. On the other hand, the fostamatinib, a known SYK inhibitor, might be also a reasonable therapeutic alternative for patients of cluster 5, where, as we've said, the type 1, the B cell responses were most prominent. And finally, probably and finally, we have also identified several compounds, novel compounds, that target a JAK-STAT pathway, and we hypothesize that those compounds might confer some therapeutic benefit for patients of cluster 3.
Yeah. So fantastic. So you identify 5, subgroups. So just lastly, in terms of your future work, what's your plan to
First of all, we need to validate our data in an independent validation cohort, and additionally, we have some other projects that we aim to use machine learning techniques in order to develop diagnostic tools, not only in lupus but also in Sjögren. And we will try to identify novel molecular biomarkers that may play a role in disease monitoring. But I'm interested, quite interested in lupus, but I find also Sjögren quite interesting.
Yeah, fantastic. Right. So thank you for offering to be interviewed by us at Room Now. So tune in to Room Now for more updates and reports throughout the Congress. So you can follow me as well at my Twitter handle at Yusof. So thank you again, Dr. Panagiotis, and all the best.
Thank you very much.
Bye-bye.
Bye.